C'est avec beaucoup de plaisir qu'on accueille Freddy HAESEBROUCK aujourd'hui, qui va faire sa présentation en anglais, qui est belge et qui est un des microbiologistes vétérinaires qui est le plus productif au monde, qui a écrit le plus de choses au monde, en particulier aussi bien sur les antibiotiques et le bétail que sur les zoonoses, c'est-à-dire les infections humaines, bactériennes, animales, transmissibles à l'homme, y compris avec des points qui ressemblent à des choses sur lesquelles on travaille, sur le cycle de la glutamine ou sur les zoonoses. Et donc le domaine dans lequel il a souhaité présenter, c'est celui des helicobacters comme agents de zoonotique. Merci.
So thank you, Professor Raoul, for this nice introduction. I also would like to thank you for the invitation and for having been so well received here. It's really a pleasure to be here in Marseille. And as already explained by the chairman, this afternoon I would like to talk about Gastric non-Helicobacter pylori helicobacters. And these are bacteria which are mainly associated with animals, with dogs, with cats, with pigs, which are also important from human health, which are important from a public health point of view, so they are zoonotic agents. But when we talk about the public health aspect and about gastric helicobacter infections in humans, the first remark that I have to make here is that these non-Helicobacter pylori helicobacters are not the most frequently detected Helicobacter species in humans. The most frequently detected Helicobacter species in humans is definitely this one, the bacterium on the left side of the slide is definitely Helicobacter pylori. But this Helicobacter pylori is a typically human-associated bacterium. So animals do not play an important role in its transmission. It is considered to be the most frequent cause of arthritis in humans. It's also the most frequent cause of peptic ulcers in humans. And it is considered to be the main cause of gastric cancer in human patients. So that's the most frequently detected Helicobacter species in human patients. However, in some patients with gastric complaints, another type of bacterium is found, what we call non-Helicobacter pylori Helicobacter species, and these are bacteria with a typical morphology. You can see it here, so a long spiral-shaped morphology, horseshoe-like bacteria, and this morphology is quite different from that of Helicobacter pylori. Helicobacter pylori has a slightly curved morphology. But it is about these long spiral-shaped bacteria that can be present in the stomach of humans that I would like to talk today. And these non-Helicobacter pylori helicobacters have also been associated with gastritis in humans. Gastritis that is mainly found in the antrum of the stomach, in the region of the stomach close to the small intestine, close to the duodenum, distal part of the stomach. They have also been associated with peptic ulcers, and they have been associated with what is called mold lymphoma of the stomach, mucosa-associated lymphoid tissue lymphoma. The mold lymphoma is a type of cancer characterized by proliferation of B lymphocytes. So you can see it here, proliferation of lymphocytes, B lymphocytes, and actually it has been described by the literature that the risk to develop that type of cancer, the risk to develop mucosa-associated lymphoid tissue lymphoma is higher after infection with these non-Helicobacter pylori helicobacters than after infection with Helicobacter pylori itself. Okay, this afternoon I first of all would like to say a few things about the general characteristics of these non-Helicobacter pylori helicobacters. Thereafter, I very briefly talk about their significance for animal health, and And then finally, I would like to consider best significance for human health or public health. But let's start with the general characteristics. So these are gram-negative bacteria, and I already mentioned that they have a typical morphology. They are long spiral-shaped bacteria. This here, this picture on the left side of the slide, that is not a gram stain, it's a Hiemstra stain, and you can see this horseshoe-like morphology here. And another characteristic is that these bacteria are very difficult to culture in vitro. They are very fastidious microorganisms. So, Gastric non-Helicobacter pylori helicobacters are fastidious microorganisms, fastidious bacteria, and actually only a few in vitro isolates from human patients are available. And the non-Helicobacter pylori helicobacter strains that are available, that have been isolated from gastric biopsy samples from humans, all belong to two species, Helicobacter visceroni and Helicobacter felis. I come back to that later. And these two species, these two Helicobacters, grow somewhat easier to culture in the laboratory than many other non-Helicobacter pylori helicobacters. So that means that you can demonstrate these bacteria in gastric biopsies from humans with other techniques, for instance by histological examination of gastric biopsy samples, microscopic evaluation of these samples, or by, for instance, PCR or other molecular techniques, but they are extremely difficult. It is extremely difficult to culture them, to isolate them. Then what about the taxonomy? This is rather complicated, and there's still quite some confusion about taxonomy and about nomenclature of these bacteria, but I usually divide them into two groups. On the one hand, there is Helicobacter suis, which is mainly associated with pigs, and then on the other hand, there is a group of 7 different Helicobacter species which are often present in the stomach of dogs and cats. And these dog and cat associated Helicobacters, canine Helicobacters, are mentioned on this slide: Helicobacter felis, Helicobacter visceroni, salomonis, how many Urohasticus, Chinohasticus, Bacillophoris. The main hosts of these bacteria. We can start with these dog and cat associated Helicobacters, Helicobacter felis, visceroni, salomonis, Urohasticus, Chinohasticus, and Bacillophoris. Main hosts here are dogs and cats, but they can also infect wild felidae, such as tigers, leopards, cheetah, etc. And from time to time they infect humans. And in all these different hosts, they mainly colonize the stomach. And then the other one, Helicobacter suis, main hosts here are pigs, but they also naturally infect non-human primates. So they also infect monkeys in nature, and from time to time they jump from pigs to humans. And again, in all these different hosts, they mainly colonize the stomach. The Helicobacter suis is highly prevalent in pigs, and the prevalence is especially high in pigs at slaughter age, and also in adult pigs in sows and boars. And here the results are presented from a study that was carried out several years ago in our department. And you can see that in this study we found prevalence of 90% in sows and boars. So in adult pigs, prevalence can be very high, and this high prevalence in adult animals, in sows and boars, this high prevalence indicates that any immune response in the pig will not result in an elimination of the infection, will not result in an eradication of these bacteria from the stomach. So in conclusion, these non-Helicobacter pylori helicobacters, those have to drive lung infections. Helicobacter suis mainly in pigs, and And then some of the species, mainly in dogs and cats, but also humans, may be infected during prolonged periods of time with these bacteria, and the host immune response, a natural host immune response, does not seem to be able to eradicate the infections of these bacteria, can persist in the host even in the presence of an immune response. Then briefly, a few things about the main virulence factors of these bacteria. And we have carried out in vitro studies in cell cultures, as well as in vivo studies, experimental culture studies in mice, in germs, also in pigs. These rodents here, mice and germs, are often used as models for human Helicobacter infections. And the studies that we have performed were mainly carried out with two species, Helicobacter suis on the one hand, and then Helicobacter harmoniae, which is dog and cat associated Helicobacter species on the other hand. So we have mainly worked with these two species, and I shall not go into detail. I try to give a brief summary of the main results of these studies. So normally these non-Helicobacter pylori Helicobacters are taken up orally. So they are taken up orally, and after being taken up orally, they enter the stomach and they arrive in the very acidic lumen of the stomach. So the first thing that they have to cope with is the very hostile gastric environment. They have to adapt to the very acidic environment of the stomach, because here hydrochloric acid is produced. And the characteristic of all gastric Helicobacter species is that they produce large amounts of the enzyme pyrase, so they are pyrase positive, and pyrase can break down, can hydrolyze pyria with the production of ammonia and also CO2, but here ammonia is important. So these bacteria are surrounded by ammonia, and that creates a microenvironment where the pH is somewhat higher, so ammonia can neutralize the hydrochloric acid. That's one thing, and then another important virulence factor is their motility. They are highly mobile, highly mobile microorganisms, and this allows them to move away, to swim away from the very acidic lumen of the stomach towards the mucosa of the stomach, which has a neutral pH. Okay, so these bacteria move to the mucosa, and there they adhere to the gastric surface, to the gastric epithelial surface, and the stomach, the surface of the stomach is covered with a very viscous mucus layer, mainly consistent of what is called mucins. And this mucus layer helps to protect the gastric tissue, helps to protect the stomach against, for instance, hydrochloric acid, but also against pathogenic bacteria, because bacteria are normally trapped in this mucus layer. They are trapped in the mucus layer. This mucus layer is secreted continuously. It is also shed continuously. It is excreted continuously, and in this way, normally the bacteria are eliminated. They are eliminated in the lumen of the stomach. However, these non-Helicobacter pylori Helicobacters are able to penetrate the mucus, and here their spiral shape morphology can be very useful, or is very useful, can be helpful. So it allows them to penetrate the mucus, to drill through the mucus, and then they adhere to the gastric epithelial cells. And characteristics are, one of the characteristics of non-Helicobacter pylori Helicobacters is that they do not only adhere to the surface epithelial cells, they also enter the glands, the gastric glands, so they reside deeply in the gastric glands. And non-Helicobacter pylori have reference for parietal cells. So So they have a preference for parietal cells. Parietal cells are the cells that produce hydrochloric acid. And that's simple for non-Helicobacter pylori, Helicobacters, they're tropismal for these hydrochloric acid producing parietal cells. So they're found in association with parietal cells, and you can see it here above. This is a chemical staining, and what you can see here, these spirochetal bacteria stained brown, or in this case, this Helicobacter suis, and you can see it in association with epithelial cells. That's translucent from microscopical picture here is parietal cells with mitochondria. So these parietal cells contain many mitochondria, which are necessary for hydrochloric acid production, which costs lots of energy. You can see close association of, in this case, Helicobacter suis with the parietal cell. So they reside deeply in the gastric glands, and they are found in association with parietal cells, and they even penetrate in the canaliculi of these parietal cells. And these canaliculi are small canals in the parietal cells, and in these canals hydrochloric acid is released. And this localization in the glands and even in these canaliculi of parietal cells may help to persist these bacteria in the host and evade the immune response. Okay, these bacteria adhere to epithelial cells, they adhere to parietal cells, and then they induce epithelial cell death, and there are several virulence factors that may play a role, but the important virulence factor here is the enzyme gamma-glutamyl transpeptidase. So that's the most important factor produced by these bacteria that is responsible for epithelial cell death and for parietal cell death. So they damage the epithelial cells, and the epithelial cell death may result in lesion development and development of ulcers. They also damage the parietal cells, resulting in decreased production of hydrochloric acid, because these parietal cells are the cells that produce the hydrochloric acid in the stomach. And we have shown this in vitro in primary cultures of parietal cells. These are parietal cells cultured in vitro, parietal cells from pigs. The nucleus is stained blue. Their ATPase is stained red. ATPase is an enzyme that is responsible, that plays an important role in hydrochloric acid production by the parietal cells. So the nucleus is blue, this enzyme ATPase is red, and then the bacteria in the cell, Helicobacter, in this case this Helicobacter suis, is stained green. And you can see here a close association again of Helicobacter suis with these parietal cells, this primary culture parietal cells. So they first adhere and then they kill, they destroy the parietal cells. This was shown in vitro, but it was also confirmed in vivo. Carried out experimental infection studies with, on the one hand, white-type Helicobacter suis, which is able to produce gamma-glutamyl transpeptidase, and on the other hand, also experimental infection studies with GGT and gamma-glutamyl transpeptidase negative mutant strain, that is not able to produce gamma-glutamyl transpeptidase. We have carried out these infections in mice, in germs, and in pigs. And after infection with the wild-type Helicobacter suis traits the expression of ATPase in gastric cells, ATPase, which is responsible, which plays a role in hydrochloric acid production. The expression of this ATPase is much lower in animals, or in the gastric tissue of animals infected with the wild-type strain than in animals infected with the mutant strain, which cannot produce gamma-glutamyl transpeptidase. And what you can see here is a parietal cells with many mitochondria, parietal cell of a gerbil, stomach of gerbil that was infected with Helicobacter suis. So this is Helicobacter suis, and this is the parietal cell showing necrosis. So they adhere to epithelial cells, they destroy the epithelial cells, and they also affect the host immune response in several ways. First of all, they can inhibit T-cell proliferation. This again is gamma-glutamate transpeptidase dependent. And then in the later stages of the infection, they upregulate the production of a cytokine. Referred to as CL13, that's a B lymphocyte, chemo attracted. So the B lymphocytes are attracted to the stomach where these bacteria are residing. B lymphocytes are attracted. There's an influx of B lymphocytes, and this may play a role in development of these acidic mucosa-associated lymphoid tissue lymphoma. I already mentioned that this is characterized. This type of cancer is characterized by proliferation of B lymphocytes, and that risk to develop this multi-lymphoma is higher after an infection with non-Helicobacter pylori helicobacters than after infection with Helicobacter pylori. This is illustrated here. What is illustrated here is a marked upregulation of that B lymphocyte chemoattractant in mice, and especially in gerbils infected with Helicobacter suis. You see an upregulation here, and this upregulation is not seen after infection of these animals with Helicobacter pylori, and that may be one of the reasons why development of the risk to develop multiple lymphoma is higher after an infection with Helicobacter suis and not non-Helicobacter pylori helicobacters after an infection with Helicobacter pylori itself. Here you can see multi-lymphoma-like lesions characterized by proliferation of B lymphocytes, multi-lymphoma-like lesions in gerbils infected, colonized during 8 months with Helicobacter suis. And again, this is more or less gamma-glutamate transpeptidase dependent, because compared to Helicobacter suis mutant strain, gamma-glutamate transpeptidase negative mutant strain infection, an infection with wild type Helicobacter suis strain induces more inflammation in this case in gerbils. So I have mentioned it several times, this gamma-glutamate transpeptidase. Then I would like to say a few things about the possible mechanism of action of this enzyme. So gamma-glutamate transpeptidase is able to hydrolyze the amino acid glutamine, can also break down glutathione, and among other things, glutamate is produced, and glutamate is very important for the bacteria because it is the main energy source of Helicobacter. Glutamine is important for the bacteria, but it is also important for the host cells. It is an essential nutrient for host cells, for epithelial cells, for lymphocytes. And when glutamine is hydrolyzed by the bacteria, it is no longer available for the host cells. This may be one of the reasons why these cells are damaged. And one thing is that glutamine is not only consumed by the bacteria also interfere with their uptake by the host cells. So they inhibit the uptake of glutamine by these host cells. This is illustrated here. What you can see here is the expression of glutamine transport protein in the stomach or in the gastric cells of mice infected on the one hand with Helicobacter suis, on the other hand with glutamate transpeptidase negative mutant strain, which they cannot produce the enzyme And you can see that these bacteria inhibit the expression of the glutamine transport protein. So the bacteria consume glutamine, also interfere with the absorption of this amino acid, and then they also hydrolyze glutathione. And glutathione is an important antioxidant for the host cells. So glutathione helps to protect host cells against oxygen radicals. It's a scavenger of oxygen radicals, and when it is broken down by the hamaglutaminyl transpeptidase of Helicobacter species, it is no longer available for the cells, so this protection against oxygen radicals is lost. And another thing is that some of the products that are produced during this hydrolysis of glutathione, some of the degradation products such as cysteine play a role in production of oxygen radicals. So you have two things. On the one hand, there is less protection because glutathione is consumed. On the other hand, more reactive oxygen species, more oxygen radicals are produced, and this may lead to lipid peroxidation of the lipids in the membrane of the host cells, and finally resulting in cell death. Okay, that's all I wanted to say about these possible virulence factors of non-Helicobacter pylori helicobacters. And now I would like to talk very, very briefly about their significance for animal health. And I start with dogs and cats. What is the pathogenic significance for dogs and cats of these non-Helicobacter pylori helicobacters that I mentioned? Actually, this is not well known. They have been associated with chronic gastritis, the chronic inflammation of the stomach of dogs and cats, and chronic vomiting. This is Helicobacter species dependent. For instance, we have shown that Helicobacter hamani is more virulent than its closest relative, Helicobacter neuromasticans. So there are 7 species that can affect dogs and cats. They are not all as virulent. There are some of these species that are more virulent than others, and the mating is very dependent within one species. Most probably there is variation between different strains in virulence. Then Helicobacter suis, pathogenic significance for pigs. We have carried out several experimental infection studies in pigs. One was trying and determine their pathogenic significance. I shall not go into detail. I try to give a summary of the main conclusions from these studies. After experimental infection of pigs with Helicobacter suis and also after natural infection, these bacteria mainly colonize the distal part of the stomach. They mainly colonize the glands zone and the antrum. So this here is the small intestine. Here we have the esophagus. So they mainly colonize the distal part of the stomach. And they also induce gastritis, an inflammation of the stomach, or at least some strains of Helicobacter suis induce gastritis, because again there is variation between different strains of Helicobacter suis, and this gastritis is mainly found again in the distal part of the stomach. It's mainly found in the antrum of the stomach. With experimental infection with Helicobacter suis also results in a reduction in the weight gain of these pigs. This may be important from an economic point of view. And then one of our conclusions was that Helicobacter suis is most probably one of the factors playing a role in what we call gastric ulcer disease in pigs. And gastric ulcer disease is clearly multifactorial disease. Several factors play a role. One of these factors is Helicobacter suis, or some strains of Helicobacter suis. A remark that I would like to make here is that pepticosis in pigs, pepticosis in pigs are not identical to pepticosis in humans. Pepticosis in humans are mainly found in the more distal part of the stomach or in the duodenum, that's in humans. In pigs, on the other hand, ulcers are almost always found in the very proximal part of the stomach. They are found in a small area, a small region of the stomach around the opening of the esophagus, which is called the non-glandular part of the stomach because it does not contain glands. And this non-glandular part of the stomach of pigs, of the porcine stomach, is very typical for pigs. You do not find such a non-glandular part in dogs. You do not find it in cats. Also humans do not have this non-glandular part of the stomach. So it's typical for pigs. It is a zone that with no mucus, because there are no glands here to produce mucus, and you can easily observe this non-glandular part if the stomach of the pig is opened here along what is called the H curvature. So when you open the stomach here, that's what you can see then. That's a normal non-glandular part. So here in the center, this is the opening of the esophagus. And around this opening, you can find this non-glandular part, which is not protected by mucus, so it can be easily damaged when it comes into contact, for instance, with hydrochloric acid. There's no mucus here because there are no glands to produce mucus. And this is the non-glandular part of the stomach of a pig that was experimentally infected with Helicobacter suis. So what you can see here are some erosions, ulcers, and you can also see what is called severe hyperkeratosis. So this is the opening of the esophagus, hyperkeratosis and erosions, and that's another case, again, non-glandular part of the stomach of a pig, experimentally infected with Helicobacter suis. So what you can see is an erosion surrounded by the zone of hyperkeratosis. So our conclusion was that Helicobacter suis is one of the factors playing a role in development of pepticosis in pigs. It's not, certainly not the only factor. But I already mentioned that these bacteria mainly colonize the distal part of the stomach. The ulcers in pigs are found in the very proximal part of the stomach. How do they play a role in the development of these lesions? It's not very well known, but most probably they may have an effect on such an infection may affect the normal microbiota, the normal microflora of this non-glandular part of the stomach, and other things because they interfere with the production of hydrochloric acid. For instance, we found very high numbers of the new Fusobacterium species in the non-glandular part of the stomach of pigs with ulcers, pigs infected with Helicobacter suis. The name that we proposed for this Fusobacterium species is Fusobacterium suis. This is a gram-negative bacterium, an anaerobic gram-negative bacterium, which probably, or it might, also play a role in development of this multifactorial disease in pigs. Okay. Let's now talk about the possible significance of these non-Helicobacter pylori Helicobacters for humans, for public health. And first of all, I want to say a few things about gastric disorders. Thereafter, I would like to talk about the possible role of these bacteria in neurodegenerative disorders in humans. But let's start with the gastric disorders. And I already mentioned that these bacteria have been associated with an inflammation of the stomach, gastritis, also with peptic ulcers, and with also associated liquid tissue lymphoma. I also mentioned that these non-Helicobacter bacteria, the non-parasitic bacteria that are found in the human stomach, can be divided into two groups. Two types. On the one hand, there is Helicobacter suis, maybe associated with pigs. On the other hand, there is this group of 7 different Helicobacter species that often colonize the stomach of dogs and cats. So most probably pigs are the main source of infection for humans, while dogs and cats may be source of infection for humans with the other non-Helicobacter pylori helicobacters. Pigs for suis, and then the other non-Helicobacter pylori helicobacters maybe due to contact with dogs and cats. How are they transferred from animals to humans? This may occur through direct contact, as you can see here. They also occur through indirect contact, through contact with vomit, for instance. And vomiting often occurs in dogs, often occurs in cats, also in pigs. Pigs with fever often do vomit. It has also been shown that these non-Helicobacter pylori helicobacters can survive in water for several days. So they also may be transferred to humans through indirect contact. And then for Helicobacter suis. Helicobacter suis might also be transferred to humans through the food chain, because we were able to demonstrate the presence of viable Helicobacter suis bacteria in minced pork, in vacuum wrapped minced pork, and also from pork carcasses in the slaughterhouse. And that might indicate that Helicobacter suis might also be transferred to humans through the food chain. That pigs and perhaps pork are the main sources of infections for humans has also been confirmed by genetic analysis of Helicobacter suis strains. So we have genetically analyzed Helicobacter suis DNA from different sources. We have carried out through the locus sequence typing on Helicobacter suis DNA from different sources. So MST was applied on Helicobacter suis strains present in the stomach of pigs from Western Europe, from the USA. It was also applied on Helicobacter suis DNA present on pork carcasses, and these pork carcasses were from slaughterhouses in Belgium. We also applied proteomic sequence typing on Helicobacter suis DNA present in gastric biopsy samples from human patients from Belgium, Australia, and Japan. We also included some Helicobacter suis strains from Asian pigs, local Asian pig breeds, and we included the Helicobacter suis strains naturally colonizing the stomach of monkeys or non-human primates. And the results are summarized here. So, very briefly summarized, all Helicobacter suis strains from gastric biopsy samples from human patients were very similar to Helicobacter suis strains present in the stomach of pigs from Western Europe and the USA. They were also very similar to Helicobacter suis DNA, which we detected on pork carcasses. So all these strains were found in one cluster. So this black cluster here is the cluster with Helicobacter suis strains from Western pigs, Helicobacter suis DNA from pork carcasses, and present in gastric biopsies from humans. So this was one cluster, and it could be clearly differentiated from for instance, Helicobacter suis strains present in some Asian pigs, and certainly also differentiated from Helicobacter suis strains that infect non-human primates. These formed different clusters. And so these results of proteomic sequence typing further indication that pigs, perhaps pork, are the main sources of infection for humans. Okay, so these non-Helicobacter pylori helicobacters from dogs, cats, from pigs can cause gastric disorders. And more recently, there are some indications that they might also affect the cause of Parkinson's disease and Alzheimer's disease in humans. And let's start with Parkinson's disease. Already several years ago, it was described in literature that in several patients with Parkinson's disease, eradication of Helicobacter pylori, so Helicobacter pylori is the typical human-associated Helicobacter species, was described that Helicobacter pylori eradication may result in clinical improvement of Parkinson's disease, but this was not shown, this was not found in all patients. There were patients that where this was not the case. So the question that was raised was, does Helicobacter suis plays a role in some, or might Helicobacter suis play a role in some of these cases, because also rural living and farm experience, and especially livestock farm experience, had also been associated with Parkinson's disease. So we tried to determine the frequency of Helicobacter suis DNA in gastric biopsy samples from human patients, and there were two groups of patients. There was a group with clear signs of Parkinson's disease on the one hand, on the other hand there was a group of control patients, patients with no signs of Parkinson's disease. All these patients were from the UK. The experimental design is very briefly here. So archived DNA extracts from gastric biopsies from human patients were tested. Already mentioned that there were two groups of patients. There were 256 patients with no signs of Parkinson's disease. So that was a control group. And then there was another group of 60 patients with clear signs of Parkinson's disease. Some of these patients also suffered simultaneously from Alzheimer's disease. And these samples were tested for the presence of Helicobacter suis and for the presence of Helicobacter pylori. And then very briefly the results. So the results for Helicobacter suis are mentioned here in yellow. We detected Helicobacter suis DNA in 2% of the control patients, and that's more or less what we expected. However, the results for the Parkinson group, the patients with Parkinson's disease, were rather remarkable. Helicobacter suis DNA was found in 27% of these patients. That is at least 10 times greater. So we found a remarkably high frequency of Helicobacter suis DNA in patients with Parkinson's disease. At this moment, we certainly do not know if there might be a causal relationship. If there is a relationship, we do not know the mechanism. To obtain better insights in this matter, we recently started a new project, a new research project. Last year we started a new project, and in this project we try to determine if a Helicobacter suis infection might have an impact on the cause of Parkinson's disease and Alzheimer's disease in humans. And actually, both diseases can be present in the same patient. Sometimes you see patients simultaneously suffering from Parkinson's disease and Alzheimer's disease. And actually, we already have demonstrated the presence of Helicobacter suis DNA not only in the stomach of patients suffering from those diseases, but also in the blood of such a patient. Both diseases are characterized by neuroinflammation and also characterized by dysfunction of the blood-brain and the blood-cerebrospinal fluid barrier. These brain barriers are very important because they determine what to enter the brain or what will not enter the brain. Okay, to try and determine if Helicobacter suis infection might have an impact and effect on the cause of Parkinson's and Alzheimer's disease, we carry out infection studies, Helicobacter suis infection studies in mice. And in one study that was carried out, one of the aims of this study was to try and determine if Helicobacter suis might, Helicobacter suis infection might induce neuroinflammation and might have an effect on the blood-brain and blood-cerebrospinal fluid barriers. So mice were already inoculated with Helicobacter suis, and in this study the animals were euthanized 1 month after infection. And very briefly, the results, we found significantly increased permeability of the blood-cerebrospinal fluid, so there was an increase in the blood-cerebrospinal fluid permeability. And we also found neuroinflammation characterized, among other things, by an increased expression of interleukin beta in the choroid plexus. So this indicates that such gastric Helicobacter suis infection may result in a disruption of this blood-cerebrospinal fluid barrier, and that it also may induce neuroinflammation. Okay, Mr. Chairman, I would like to end my talk with some general conclusions. So we have seen that these non-Helicobacter pylori helicobacters are long spiral-shaped bacteria, that there are 2 types of non-Helicobacter pylori helicobacters that can be present in the stomach of humans. On the one hand, there is Helicobacter suis, mainly associated with pigs, and then there is the group of 7 different Helicobacter species associated with dogs and cats. Pigs on the one hand, dogs and cats on the other hand, may be a source of infection for humans. Virulence factors of these bacteria, several virulence-associated factors have been described, but an important virulence factor is the amyloid transpeptidase, which in modern days can induce bacterial cell death and rifle cell death. Then the pathogenic significance for dogs and cats is not very clear yet. They have been associated with chronic arthritis and chronic pathogenic significance is Helicobacter species dependent for dogs and cats. Pathogenic significance for dogs and cats is Helicobacter species dependent, may also be strain dependent. Then in pigs, Helicobacter suis has been associated with arthritis, with increased weight gain. It is one of the factors playing a role in gastric ulcer disease in pigs. Non-Helicobacter pylori helicobacters are zoonotic agents. They have been associated with gastric disease in human patients, arthritis, peptidosis, mouth, lymphoma. And then with regard to the possible impact of these non-Helicobacter pylori helicobacter species on the cause of neurodegenerative disorders in humans, we found remarkable high frequency of suis DNA in gastric biopsies of Parkinson's disease patients, and in mice, connection to lung infection of mice with Helicobacter suis causes neuroinflammation and it discharges the blood-cerebrospinal fluid barrier. With regard to the possible impact of such an infection on neurodegenerative disorders in humans, there remain a lot of questions, and actually there are more questions than answers. We are not sure at this moment whether these infections may indeed have an effect on the course of these chronic neurodegenerative disorders in humans. That's the subject of further studies. These are the people working in our department. Several of them are involved in Helicobacter research. Then the studies on Parkinson's disease in human patients were done in collaboration with a group of Sylvia Dobbs and John Dobbs at King's College and Wolseley Hospital in London in the UK. And then our studies on liver inflammation are done in collaboration with the group of girls from the the Inflammation Research Center in Ghent, at Ghent University. And Mr. Chairman, ladies and gentlemen, I would like to thank you for your attention.
I'm really interested in the gamma-glutamine transpeptide story. So, I get the question, is there any other pathogenic bacteria that have this enzyme? And the second, do you suggest that maybe peptic booster in humans should be treated by glutathione, for example?
With regard to the first question, there are indeed several other bacteria that produce this enzyme, and also eukaryotic cells produce glutathione, produce this enzyme. It's not completely the same. So when you look at amino acid level, it's not completely the same, but there are other molecules that produce it. We don't know if it's in the same amount that they produce it, but it is produced by several other bacteria. And then you said the second question, can we use this enzyme to treat humans?
Glutathione.
Glutathione. Sorry, can we use glutathione? Actually, we have carried out studies in germs infected, in this case with Helicobacter suis, and some of the animals were treated with glutamine, others were treated with glutathione, and then there was a control group. In the animals treated with glutamine, the inflammation is much lower, most probably because then glutamine can be used by the cells. Then with regard to glutathione, there you have to be careful. It depends on how much glutathione that you have. When you have small amounts of glutathione, the bacteria will produce more cysteamine glycine, which can have a negative effect. When you have large amounts of glutathione, but for animals that will be very cheap, very expensive, then it will be cheap. When you have large amounts of glutathione, then they will be protected. Because on the one hand you have a protective effect, on the other hand you have the helicobacter that destroys, that breaks down glutathione, the production of some of the degradation products that can induce production of oxygen radicals. So there it's rather tricky.
Is there any data regarding bismuth? Is there any data regarding the action of bismuth with non-Helicobacter, non-pylori, sorry?
Well, it's used for treatment of, normally you have triple therapy in humans, 2 antibiotics and a proton pump inhibitor. Sometimes also bismuth is used, which has, can have a negative effect on Helicobacter pylori. It has not been, as far as I know, it has not been tested in but I suppose that it will have the same effect.
When we're doing the breath test for the diagnosis, since the non-H. pylori helicobacters are erased positive, they will give us a positive reaction. So when our diagnosis patients, we will just confirm that they have H. pylori, and we might be in the case of non-H. pylori helicobacters. In this case, the treatment used for the eradication of H. pylori works for these helicobacters also.
So the question is, can you use the same therapy for a non-Helicobacter?
Since the test is also positive, the breath test for this bacteria are also positive. Since they
you mean the diagnostic test? Or?
Yes.
Okay, yeah. For diagnosis of Helicobacter pylori, Helicobacter pylori infections in humans, what is used can be, you have the urea breath test, because these bacteria produce large amounts of urease. So in this case, the patient receives a drink with labeled urea, C13 or C14 labeled urea, and then this is the labeled CO2 that is measured in the breath. You can try to use this for the non-Helicobacter pylori helicobacters, but it will often be negative. And the reason for this is that these bacteria cause very focal infections, very local infections, very patchy. The infection pattern is very patchy. It's also often located in the distal part of the stomach, and that's one of the reasons why the raised breath test may be negative. So the diagnosis is mostly, if the diagnosis is made of these infections, it's done by arthroscopy, RC biopsy is taken, but there also you have to be careful because, again, due to this very patchy colonization pattern, even when you take a sample, and then normally it's, they use histology, they also can isolate Helicobacter pylori. These non-Helicobacter pylori helicobacters cannot be easily isolated, so you need to do histology. It can be false negative because you only take a very small sample, so a section of 4 micrometer. And we had a veterinary surgeon with severe arthritis. So we received samples. We demonstrated the presence of Helicobacter suis. We could clearly demonstrate it by PCR. And we also carried out histological examination. And in some glands, in some sections, you found large amounts of these roach parasitic bacteria in the glands, but on other sections we didn't see anything. So the diagnosis is not easy. Okay. Was that an answer to your question?
Okay, thank you very much.